Hello and welcome. The following videos are 5-minute talks created by participants in the Yale BBS Diversity and Inclusion Collective Science Communication Series. Each scholar worked with mentors from the graduate student organization, Yale Science Communication, to design a talk on any topic of their choosing. The talks have been organized into 5 broad categories: neuroscience, cell biology and genetics, human health and disease, human evolution, and science policy. If you're interested in a specific talk, you can navigate to the appropriate timestamp. At the start of each section, the talks in each section will be listed with their appropriate timestamps. We hope you enjoy these talks and congratulate the scholars on their hard work.
Hello everyone, my name is Dorian Padilla and today I'm going to be talking about really awesome cells called microglia and how they are considered the fighter against nervous system diseases. And so the first question we should be asking ourselves is, what are microglia? And if we take the word microglia and separate it, we have 2 words: micro, which means something that is extremely small, and glia, which is the supporting tissue of the brain consisting of different cell types. Among those cell types, we can find microglia. Microglia are only located in the brain and spinal cord. They account for 10 to 15% of all the cells in the brain. And most importantly, they are the soldiers of our brain. So let's take a time to review the functions or responsibilities of soldiers. In times of peace, a soldier will spend their career improving their skills in training, just as microglia do in times of peace. They grow, they train, and they prepare for any foreign attack. Soldiers patrol areas for security or to seek out the enemy, just as microglia do in the brain and spinal cord. They are not static cells. Soldiers conduct military attacks and defense, a common function they share with microglia, because microglia attack pathogens that enter the brain by phagocytosis, which is a process where the cell goes out and takes in or eats a specific structure to be degraded. Finally, but not least importantly, soldiers escort and engage in protective duties, just as microglia do in the brain and spinal cord. And these protective duties have actually made microglia cells stronger to fight certain diseases from the nervous system. One of them being Parkinson's disease, which is defined as a nervous system disorder that affects movement. Mad cow disease, which is a fatal nervous system disorder believed to be caused by an abnormal protein called prion. And the focus of my presentation, which is Alzheimer's disease. Most of you may have heard about Alzheimer's disease because this disease is one of the most common progressive brain disorders that slowly destroys memory and thinking skills. As you can appreciate in the image in the left side, we see a healthy portion of the brain, in comparison with the right side, where we see an advanced Alzheimer's brain. And we can clearly see that some of the brain structures have grown and others have shrunk. At a molecular level, there are specific proteins that are called amyloid beta, and these proteins are the main ones responsible for the progression of Alzheimer's disease. And these specific proteins can form plaque, which is a ball of protein or a cluster of protein that is insoluble in the brain and can be really difficult to eliminate from our bodies. And if it's really difficult to eliminate from a body, it can be really toxic for the brain. Imagine this amyloid beta plaque as one of your headphones being tangled up. Well, I know that some of us have had difficult times untangling those headphones. Well, some of the cells found in our brain have had difficult times untangling amyloid beta plaques. That is why microglia are so important, because microglia, it has been shown that they can eliminate amyloid beta plaques. And they do this with the help of some certain proteins that are found in the cell membrane of microglia. And these proteins help microglia eat this ball of proteins by facilitating the process of phagocytosis. And as we can appreciate more in the image, in the microscope image, we can see that microglia are represented in white, amyloid beta plaques are represented in blue, and the protein that I mentioned before is represented in red. And we can see that specific interaction between microglia and amyloid beta plaques. where we can see that that protein in the cell membrane of microglia is the one that is recognizing the amyloid beta plaques and helping microglia degrade it. So to summarize what we have learned today about microglia, they take care of the brain and eliminate infectious agents such as virus. They recruit other cells for other functions in the brain, such as repairment of the brain tissue. And they protect us from neurodegenerative diseases, such as Alzheimer's disease. Here are the references. Feel free to pause the video if you would like to learn more about microglia. And with that being said, I hope you had a wonderful time learning about microglia. Thank you.
Well, hello, my name is Francisco Torres from the University of Puerto Rico at Cali. Today, I'm going to be teaching you a little bit about a substance called myelin and its role in the nervous system. I hope you find this presentation informative. Firstly, it's important to remember that the nervous system is made up of these specialized cells known as neurons, and these communicate with each other using electrochemical signals. Once a neuron or multiple neurons stimulate another neuron enough, this may form an action potential. Action potentials are essentially electrical currents that run through specialized tail-like structures known as axons. You can imagine the electrical current running down an axon, much like a car runs down a road. But how do these action potentials actually form? Well, they form through a process called depolarization. Essentially, currents are formed when negative charges flow through a system. And in depolarization, a positive charge is seen to flow inward while a negative charge flows outward. Now, in unmyelinated systems, the whole of the axon must be polarized for the charge to run through it. To use our analogy, once again, this would be like a car being stuck in traffic. You can only move forward once the car in front of you moves forward. In the same vein, the current can only move forward when the depolarization occurs in that segment of the axon. So different animals have evolved different strategies to speed up the transmission of the current. And to understand this, we could take a look at the evolutionary history of the nervous system. For example, one important step that occurred in the evolution of the nervous system is cephalization. As animals became more mobile and needed to integrate more information from their environment, then the nervous tissue that integrates that information and processes it concentrated on the front end of these organisms. As you can see between the step between a hydra and a planaria, the difference is very drastic. Another key transformation or evolutionary step in the animal kingdom that we can study is that of giant axons. Squids have giant axons, which are much like regular axons, though the key difference is that they have a much larger diameter. This larger diameter means that the current can travel at a much faster rate than that of regular axons. Conversely, in vertebrate organisms, myelin coats, or a substance called myelin coats this axon. So instead of having an axon of a much larger diameter, the substance acts as an insulator to maintain the current within the axon without it having to depolarize completely. Note that in the image on the left, the axon is not completely coated by the myelin. Instead, a myelin sheath only covers certain segments and leaves a certain segment exposed. This is known as a node of Ranvier, and these are the sites of depolarization within the axon of myelinated systems. But how does myelin actually work to speed up the transmission of current? Well, as I mentioned, it is an insulator. So if you note in this GIF, myelin covers certain segments of the axon, so these segments do not have to depolarize. Instead, you can see that the current jumps between those nodes of Ranvier, and these are the only sites of depolarization in the axon. So this is saltatory conjunction, and to use our analogy, once again, this would be like a car racing down a road without needing to wait for other cars in front of it to move forward. Instead, it can just go as fast as it needs to. Another detail I wanted to talk about regarding myelin is that its formation varies across the nervous system. In the central nervous system, that is in the brain and in the spinal cord, myelin formation is taken care of by these glial or helper cells known as oligodendrocytes, while in the peripheral nervous system, that is the nerves that innervate the rest of the body, myelin is taken care of by Schwann cells, which are another type of glial cells. Excuse me. Now that we understand the importance of myelin and its formation, then we can ask ourselves what happens when myelin doesn't function properly. And we could study this using cases or diseases such as multiple sclerosis, where it happens just as I said. Myelin formation or myelin degrades in the nervous system. And to use our analogy once again, this would be like a car or running down a road full of potholes. The destination might not even be reached by then, and the car would at the very least be slowed down. In this case, or in the case of the axon and the current, then the current might not even reach its destination at the axon hillock. Now, since myelin is so ubiquitous or widespread within the nervous system, people that suffer from MS have a wide variety of symptoms that include cognitive, sensory, and motor dysfunction. So you can tell that this disease is terrible. It has a wide variety of symptoms. So it's important for us to understand myelin formation and degradation so that we can research this disease and find potential cures. To conclude, we studied how animals have sped up their transmission of current through different strategies. Squids have giant axons, which inhibit or decrease the internal resistance within their axons. Meanwhile, vertebrates use a substance called myelin, which insulates those neurons and helps speed along the current around the axon road. And these are my references. I hope you found this presentation enjoyable and informative, and I hope you have a nice day.
Hi everyone, my name is Leanne Vasquez. I am a scholar at the YBDIC program, and today I'm going to be talking about how autism molds the brain. So to get a little bit of a sense of what I'm going to be talking today, I'm first going to start by defining what is autism spectrum disorder. Then I'm going to go ahead and talk a little bit about the common brain structures that have been associated with, and also the scientific findings that have been related to those structures. So what defines the autism spectrum disorder? Well, actually, autism is a group of neurodevelopmental disorders that have been characterized by having repetitive behaviors, impaired communication, and also impaired social behavior. And so there are certain factors that have been suggested to play a role in its pathogenesis, such as genetic, immunological, and environmental. And it's these factors that are contributing to distinct structural changes in the brain of these patients. And so that's what I'm going to be talking about next. So there are certain areas that scientists have been able to find that have been very common, such as the cerebellum, which is known for motor coordination, the hippocampus, which you may have heard that is known for learning and memory, the amygdala, which is known for the emotional responses, and the cerebral cortex, which has many higher cognitive functions. And so today I want to focus more on the cerebral cortex due to its relevance to social behavior and communication. And so how do we know all of this? Well, it's actually the work of scientists. And so a group found a decreased cortical thickness in several regions of the brain. If we take a look to the right, we see an MRI result that was analyzed with some software. And if you take a look at these red spots in the cortex, these are the areas that they were able to find a reduced cortical thickness. Interestingly, with these same results, they were able to correlate that reduced thickness with higher social impairment, according to the scores of the Autism Diagnostic Interview Revised, which is a standardized interview used to diagnose autism. And so if we take a look at this graph here below, this is an area specific of the cortex. They were measuring on the y-axis the cortical thickness, and on the x-axis the ADI social score. So as the cortical thickness was decreasing, the ADI social score seemed to get worse. And so even more interesting, these same group of scientists were able to find an increase and decrease in cortical thickness, but in a different region. And so these were also correlated with greater communication impairment, which is another characteristic of ASD patients. So let's take a look at these two graphs again. They were measuring the same thing, cortical thickness, but this time the ADI communication score. So as the cortical thickness increased in these areas in specific, the ADI communication score seemed to get worse. And on the same hand, if we look at the graph on the below, in these areas in particular, as the cortical thickness decreased, the ADI communication score again seemed to be getting worse. So we're kind of sensing here or seeing a very similar characteristic, but a different pattern. So what does this tell us? What does this mean? Well, actually, it's because autism is a heterogeneous disorder. The many subtypes that it has and the other factors that are contributed can be explained by the different neuroimaging results. And so future research should actually be focused on developing new advanced techniques that can help interpret or elucidate in more detail these different patterns of abnormalities. And so that's why it's so important to research the different subgroups in order to develop specific treatments for these patients, which is what science is all about. That is all I have for you guys today. Thank you so much for watching.
Hi, my name is Carla Rivera, and today I'm going to be talking about creating neurons to create memory. Well, this is your brain. It works hard every single day to help you do many, many things, such as feeling, thinking, seeing, and even breathing. But one of the things we associate our brain with most is its ability to remember. Well, have you ever sat down and wondered, just exactly how can my brain remember new things? Scientists many years ago were also asking themselves this question. And through the many different answers that they found that it keeps growing. Well, specifically, one part of your brain keeps growing, the hippocampus. This small seahorse-shaped part of your brain helps in many different functions, such as emotional behavior. It becomes your own little Google Maps because it helps with spatial navigation. And it also works very hard in memory and learning through two different ways. It can be the middleman in a process known as memory consolidation, or it can also be your own little neuron factory. Well, this is a neuron right here. They're the basic cells of your brain and your nervous system. They basically fire up at each other and talk to each other constantly. They help you to move and to breathe, but they also join up so that you can think and remember stuff. Now, neurons are formed in a process that scientists call adult hippocampal neurogenesis. And while this name might sound very long, what it basically means is that once we become adults, we have these things called stem cells, and they will keep growing and maturing until they can form new cells. In our brains, they start off as a neural progenitor, which will then keep growing and moving towards the parts that your brain will need it to, and it will pass through neuroblast and become a neuron. And then it can join up with all the other neurons to help you remember. Now, what exactly do these new neurons do? Well, scientists think they can do one of three different things. They can help you through pattern separation. Can you see the difference in these two images? That is pattern separation right there. And basically, these neurons just help us remember the differences between things. They also give our memories temporal context, which is mostly used when we have memories that are very near to each other. So let's say you had breakfast at 8 a.m. and then you had like second breakfast at 8:30. Yeah, you will remember the difference between them thanks to this temporal context. And finally, they also help with memory resolution, which is basically just how clearly can we remember things. Now, what exactly does happen if these new neurons stop forming? Well, what scientists have found is that your neurons will stop forming through two ways, either just normal brain physiology, which is basically aging, or illnesses such as anxiety and depression, or it could happen through a process known as neurodegeneration, which is basically when your neurons lose their function over time. And it mostly appears in diseases like Huntington's, Parkinson's, and Alzheimer's. Now, the way in which the loss of neuron affect our brains is very noticeably seen in two main aspects: our cognitive involvement, or ability to think and remember, or our mood, or our ability to feel in different ways. As you can see in this chart, no one thing leans fully towards mood-related symptoms or towards cognitive involvement. because these new neurons are important for both of them. It has been shown that people with depression also forget things easily, but it's also been seen in the Alzheimer's patients that they develop depression and anxiety as their illness symptoms move along, because these new neurons are necessary for our brains to normally function. I hope you guys learned a lot with me today. Have a nice day, everyone.
Hi everyone, my name is Rashay Roberts and I'm an undergrad at the University of Maryland, Baltimore County, currently doing research under the supervision of Dr. Tara Gates. My topic for today is that examining reward-related behavior is important to understanding mental health. The overall goal of the lab is to investigate the brain wiring involved in depression and other psychiatric disorders. What we know in the scientific community is that dysfunction of the circuitry, so the specific pattern that these neurons communicate to each other, contribute to psychiatric disorders. And even though this is known, we do not know the specific circuitry that relate to the specific psychiatric illnesses. So we're hoping that by determining that we could identify potential therapeutic targets in order to treat these psychiatric illnesses. So my topic for today will investigate social reward as it relates to contextual reward-seeking behavior. And this just means associating your environment with a reward. And we know that this behavior is important in animals in the wild for survival. And it's also present in humans as we go through life. We make a note consciously or unconsciously of the things that make us happy. And we either seek out these things or we avoid the things that make us unhappy. So in order to do the study, we'll be looking at a particular brain region, two brain regions, and we'll be investigating two different circuits. So the red circuit has been implicated in spatial and contextual learning, and the green circuit has been implicated in emotional regulation and stress response. And even though these are two distinct circuits, we hypothesize that they work together to contribute to this contextual reward-seeking behavior. And this is important in studies of depression because when we're looking at social reward, one symptom of depression is that you'll see people stop hanging out with their friends and they'll isolate themselves instead. And this is also present in animals. So we're going to be trying to model this in the mice model. So like I mentioned before, the overall goal is to assess the behavioral changes when the circuit is disrupted. So what I'm going to be doing right now is setting the baseline behavioral analysis before there's any disruptions in the circuit. So going forward, I'm going to be trying to answer the question of, will mice associate an environment with social interaction reward? So how did we do this? So like I said, we're trying to get these mice to make an association between their environment and the reward, and we'll be using social reward in the form of interaction with another mice. So we created two different environments. As you can see, we have a blue and green chambers. The blue chamber has dotted walls, while the green chamber has striped walls. And what we're going to be doing is placing a reward in a specific type of chamber, and then the other chamber will be absent of a reward. So I'm just going to go through a scheme and show you what this might look like. So the first day, we might place the mice in the spotted chamber with the reward in the morning, and then in the evening, we'll place them in the striped chamber without the reward. And then the next day, we'll counterbalance it by starting off the mice in the striped chamber without the reward, and then placing them in the evening in the dotted chamber with the reward. And we're hoping that if social interaction is rewarding enough, then the mice will remember which chamber they found the other mice and will revisit it and spend a much longer time there compared to the chamber without the reward. So what did we find? The chart to the left is demonstrating the percent time that the mice spent in the social chamber, both before and after the reward. Like we said, if we are expecting the social reward to be rewarding enough, then they will increase the time that they spend in the social chamber after they've been introduced to the reward. And our results, unfortunately, do not show any statistical significance And this begs us to ask a few questions. The first is, did we give the mice a long enough time to make this connection between where they found the reward and where there was no reward? So in the future, we might want to increase the length of conditioning. And then the other question is, was social interaction rewarding enough to activate the circuit? And in order to test that, we're going to try using food as a reward instead. And then another future goal is to connect back to the overall goal of the lab, to revisit the disruptions in these circuits and see what behavioral changes arise as a result.
Hi, this is Shom from India, and today I want to talk to you all about a very fantastic biological phenomenon called mitosis that happens within our body, and small tiny cells called neutrophils, they actually act like Spider-Man, which shoot out their own DNA like webs and try to catch the bad pathogens that are trying to invade our body. So the key takeaway from this talk would be that neutrophils, they cast a net which comprises of their own DNA to catch the pathogens that are trying to invade our body. So let's see how this happens. But before we get into that, we must understand what are neutrophils. So this is what a neutrophil looks like. And you can see the nucleus here, which is multi-lobed. And you can see the small, tiny granules scattered all through the cytoplasm of the neutrophil. So essentially, the neutrophil is an immune cell. It's a special type of cell called the white blood cell, and they are called granulocytes because of the tiny granules that are present in them. And these neutrophils are actually like our very own Spider-Man, because whenever a bad bacteria or a pathogen tries to invade our body, the neutrophils act like the first responders which arrive there and try to defend our body and confer immunity upon us. What the neutrophils do is a very special phenomenon called mitosis, where you can see a certain acronym called the NET. NET stands for Neutrophil Extracellular Trap. Here is a figure that is showing how the net formation takes place. So essentially, the neutrophil is the Spider-Man, and the DNA that usually remains within its nucleus is actually thrown out of the cell like a net, which goes and captures the pathogen, which is the bad guy, immobilizes it, and then kills it. So we shall take a look at how this happens at greater details. So what exactly happens is, when a neutrophil goes to the site of infection and encounters a pathogen, there are certain enzymes that are present within the neutrophil, which generates a lot of reactive oxygen species. And these reactive oxygen species, or as they are known as ROS or ROS, are not good for the cell. They cause the granules to break up, and that releases a certain protein called the N protein into the cytoplasm. Once the N protein comes into the cytoplasm, it then goes to the nucleus and attaches to the DNA in the nucleus. The DNA inside the nucleus remains in a very tightly condensed form. But when the Ne binds to this DNA, because of the DNA protein interaction, the DNA begins to decondense. And it slowly expands and it covers the entire neutrophil. And finally, the neutrophil just bursts open and it sends out its own DNA like a web, and it captures the pathogen, immobilizes it, and then kills it. This is the whole phenomenon of mitosis that we just saw. Now, I will actually show you all a short video where a scientist is actually going into the cell to see the phenomenon of mitosis. So as you can see, these cells are basically neutrophils, and the blue stain is the DNA that is condensed and present within the nucleus of the neutrophil. But during mitosis, what happens is the DNA is thrown out. And here you can see the green spots, which are basically the protein that is tagged with a green dye that is present with the net. And this is the formation of the net, or a phenomenon that we call as mitosis. Moving forward, I would like to conclude the talk where we learned about neutrophils. We learned that these are white blood cells, which are also called granulocytes, and they help us fight pathogens. One of the mechanisms that they undergo is actually called mitosis, where NET stand for Neutrophil Extracellular Trap. And the reason why I thought that this is a very interesting phenomenon to talk about is because the DNA usually remains inside the cell. But here, the neutrophils are throwing out their DNA. They are sacrificing themselves, just like Spider-Man would probably do. And that is very interesting to look at. Thank you.
Hi everyone, my name is Gladys. Today I'll be talking about my presentation titled Policing Your Immune System of Regulatory T cells, which are one of my favorite immune cells. So within our body, we have our immune system that protects us from any bacteria and any infections that might arise. And so we have our innate response as well as our adaptive response. And our innate response, all these cells are one of the first to go into action whenever we have any potential infection. And then these adaptive cells to the right are usually take a little bit longer to act and usually help maintain a long-term protection. And so today I'll be talking about one of my favorite cells called regulatory T cells, which are a type of T cell. And so I like to think of regulatory T cells as the policemen of our immune system. And so if you imagine being on the road, police officers usually are very important in maintaining constant traffic and preventing anyone from going at excess speed and potentially causing any damage. And so in the same way, regulatory T cells can help maintain a balance in our immune response and help do this by helping suppress other immune cells that may be overly reactive and preventing them from being reactive when not appropriate. And so because regulatory T cells are a type of T cell, they are initially found in what we call the thymus, which is here on the left, and it's an organ found right in between your lungs, and it's very important in the immune cell development and is a place where immune cells go and kind of specialize into a variety of cells that you saw on the first slide. And so in order for these cells to become specialized and become activated, they need the help of antigen presenting cells. And for example, I have here on the right the dendritic cell in yellow, and the dendritic cell is important because it has these really nice long finger-like structures where it can survey the environment and feel for different what we call antigens, like this little red dot, and present it to a T cell. And that way, if it does so in the correct two-step process, it can then be activated, and this T cell can go into develop into regulatory T cells, for example, or the other cells. And so these regulatory T cells use their specific surface proteins to suppress other cells and have their function. And so regulatory T cells are known to have CD25 as well as CD4 on their surface, and then express this protein called FOXP3. And these are important for maintaining their function, and FOXP3 in particular helps them divide and make copies of itself, and in that way also produces signaling molecules called IL-2. And these are some molecules that help in suppressing other immune cells. And regulatory T cells are important in the field of autoimmune diseases and transplantation. And so one example I wanted to talk to you about today was graft-versus-host disease, or GVHD, which affects patients who get a bone marrow transplant many times to help cure blood cancers like leukemia. And so these patients end up getting a whole new immune system from a donor. But in the case of GVHD patients, these immune cells end up being overly reactive and attacking the host or the patient. And so this leads to them having damage to their organs and their skin and being harmful to them. So because they have an overreactive immune system, cells like regulatory T cells can be very helpful. And so because of this idea, researchers have tried looking into how this can be modeled in mice. And so this figure shows percentage survival of mice who have GVHD in the dotted line, and then mice who had GVHD but were later given treatment with regulatory T cells. And as you can see, the percentage of survival, the percentage of mice who survived significantly increased when they received the treatment in comparison to those who did not. And so this can be potentially show the benefits of these types of cells and the potential use of these in humans. And so I just want to leave you off by reminding you that regulatory T cells act like the policemen of our immune system. They help our immune system be in check, just like policemen do in maintaining traffic. They also suppress other reactive immune cells. They have specific characteristics like FOXP3, CD4, and CD25 to help them interact with other cells and effectively suppress other immune cells. And they're very important in preventing autoimmune diseases and allergies and graft-versus-host disease, like I mentioned. So thank you very much for your time, and hopefully you found these cells very exciting as much as I do.
Hi everyone, my name is Maritza Lopez. Today I will be presenting the impact of HeLa cells in modern medicine. The innovation of cell culture research has been well underway because of an African-American woman named Henrietta Lacks. Henrietta Lacks was born in Clover, Virginia, and in 1951 she was treated by Dr. Guy, who diagnosed her with cervical cancer. Since then, her cells have been used to grow more cells for research innovations. Her cell line was known as the first immortalized human cell line and one of the most important cell lines in medical research. Right, so we're looking at how the HeLa cells innovated science then and now. So in 1951, HeLa cells were cultured for research, and then in 1955, HeLa cells were used to understand the virus called poliomyelitis, and that led to creating the polio vaccine. And then fast forward to now, 2021, HeLa cells were used to find the infectivity with the use of a molecule called ACE2 to find the spread connection of COVID-19, which we will look more into next. These are the HeLa cells affected by polio, and it's showing the effect of the antibodies. If it weren't for HeLa cells, more than 17 million people would have been paralyzed by polio. So here we are looking at the HeLa cells with the COVID-19 findings. So regular HeLa cells did not have the proteins to attract the SARS-CoV-2 virus particles, but with the ACE2 gene, the virus was found to infect and show the spread of COVID-19. Because of the HeLa cells, we understand the infection with SARS-CoV-2 particles on the COVID-19 virus. So why do the HeLa cells matter today? So at the time, the doctors who took the cells for research did not let Henrietta Lacks know, nor her family know, that they were taking her cells for research. So no consent was given whatsoever. So this brought the application of ethical guidelines to come to life, and it became administered so that other patients wouldn't be stripped of this privacy. HeLa cells revolutionized the process of commercialized cell culture and has affected medicine in the past and to this day. Thank you.
Hello, I'm Jacob Pena, and today I will be presenting on cellular communication and the conversation within you. How many of you try to eat healthy or take vitamins? Or how many of you have even wondered why you do or why you're told to? Well, here we can see a graph that's between the binding rates of, well, cellular communication in time, in seconds, of course. So we see a steep drop off in graph A as we approach the 100-second mark, whereas in graph B at the 300-second mark, we start to see a gradual drop off. The difference between these two is the presence of calcium ions, which calcium is a very vital part of a balanced diet, of course. So A, having the lack of calcium ions, shows a steep drop off in cellular communication, while B shows the healthy, steady drop off of cellular communication due to due to the burning up of the calcium ions as cells do. Thus, we can start to see a correlation between cellular communication and what we consider a healthy lifestyle. So, why do we need to be informed on this? Their role in sustaining life is unparalleled, as what would you be if your cells couldn't communicate? How would you be able to construct a skeleton, able to withstand the pressures of life? Or even how would you be able to undergo the processes of life, like rudimentary movements and even your heart pumping, as in both cells are needed to communicate. So to accomplish this, there are many means of cellular communications, and these come in the form of paracrine, autocrine, endocrine, and signaling by contact. The most basic of which is signaling by contact, as this is the equivalent of tapping someone on the shoulder or even speaking to them in person, as a cell creates transports only to cells next to them that transport messages from one cell to the other, instructing the neighboring cell on what to do to change to be more fitting towards the tissue. Now, this can only happen to a cell next to it. So what happens when we need to communicate with a cell that's on the same tissue but farther away? Well, this is where paracrine signaling comes in. Which is the equivalent of trying to get someone's attention in a crowd where everyone receives the message, but only one person or someone responds. In this form, a cell releases the messengers into the general environment in which only one cell can respond to the antigen, as it's only formed towards that cell. Just as calling someone's name in a crowd typically only gets a response from them. Thus, only the targeted cell will respond to this. Though we must stretch further to maintain life, and this comes in the form of endocrine signaling, in which case this is contact between tissues, which is the equivalent of phone calls or texting between cell phones. So in this, a cell can release hormones or messengers through the bloodstream until it reaches another cell and makes its message known. Thus, different tissues or even different organ systems can communicate with each other, thus synchronizing them and making each other's presence known. So next, we must see how a cell communicates with itself through autocrine signaling. So this is the equivalent of getting a notification on your phone or whatever you get notifications on about something that you forgot of that you need to take care of. So in this, the cell sends out a messenger, and eventually it will come back to the cell and remind it of a change it needs to undergo, in which case the cell will then undergo that change and return to its normal status. And this can go on and on and on until it's finally balanced. So, might I ask, what happens when cells can't communicate, or even further, for a longer period of time? This is when life starts to go wrong, as diseases can occur due to this lack of communication, or even miscommunication due to protein mutation. So these can result in diseases such as osteoporosis or cholera or diabetes, multiple sclerosis, Alzheimer's disease, and heart disease, and to an even extent, cancer, as cancer can result when cellular communication is completely lost between two cells. And yeah, So in conclusion, cell signaling is really vital towards the processes that maintain life. And interestingly, just as we do, cells communicate in various forms, just as we have various languages, and none are necessarily more important than the others. That's a vital fact. And cells' ability to communicate are dependent on the lifestyle we live, as the food we intake or our activity levels all play a role in it. Thank you.
Hello, everyone. My name is Amanda Anqueira Gonzalez, and today I will be talking to you about gene expression, a process that is critical for life. So what is gene expression? Gene expression is defined as a process by which the information in the DNA of a cell is used to direct the assembly of a protein molecule. So what is DNA? Think of DNA as your grandmother's old cooking book. Just like a cooking book, the DNA in our cells holds the instructions or recipes to make different proteins. These different recipes within the DNA are called genes. When a gene is made into a protein, it is called gene expression. In order to make a protein, cells can't just rip out the gene they want to express. If you want to make a recipe from the cooking book, you don't cut out the recipe, you copy down the recipe on a piece of paper. Our cells do the exact same thing. Cells copy down the information from a gene onto a molecule called mRNA. Further on, mRNA in the cell is read and made into a protein, just like you would make food from a recipe you copied down. It doesn't stop there. You can use your grandmother's cookbook to make different recipes. You can even create different restaurants with the same cookbook. Take, for example, an Italian restaurant and a Mexican restaurant. They look very different from each other, and they serve very different food. These restaurants make different recipes, although they have the same cooking book and ingredients. What kind of restaurant they are depends on the recipes they choose to make. This also happens to the different cells in our body. Cells make different proteins, which gives these cells an identity, what makes them different from each other. Take, for example, this purple mRNA molecule, which creates a red blood cell. This white mRNA molecule creates a nerve cell. And in the case of this orange mRNA molecule, it creates a muscle cell. Since proteins are the machinery of the cell, we need proteins to be made correctly and in the needed amount. Having too much, too little, or incorrectly made proteins can cause problems. If everything goes correctly in this gene expression process, our cells are healthy and happy. However, if anything goes wrong in this process, our cells become sick and unhappy. Disruptions in this gene expression process can lead to problems and disease. Here are some familiar examples of diseases that can be caused by disturbances in gene expression. By way of example, in sickle cell disease, just one protein is incorrectly made, which causes cells to go from this happy, healthy, and round red blood cell to this unhappy, sick, and skinny red blood cell. So now you see how gene expression is not only key for correct cell function, but for cells to have identities. Now you know that gene expression is critical for life. It keeps you and your cells healthy, happy, and it makes you, you. Thank you so much.
Hi, my name is Marcella, and I'll be presenting Epigenetics and Trauma, the Missing Link. So trauma is a deeply distressing or disturbing experience. And some effects of trauma are emotional dysregulation, so inability to regulate emotions, dysregulation of cortisol levels, which is the stress hormone in the body, hyperarousal. So for example, you may react more strongly to a balloon pop, increased risk of physiological diseases. So trauma has been linked to increased risk of diabetes, cancer, heart disease, and increased risk of mental disorders, so such as PTSD, depression, anxiety. So there's a growing body of evidence that offspring of trauma survivors are more likely to experience effects of trauma. They're more likely to experience higher rates of depression, PTSD, metabolic disorders such as diabetes or heart disease, and even premature death. So this figure is showing it's descendants of survivors of the Armenian genocide. So it's measuring the PHQ-9 score. So the PHQ-9 score is a depression questionnaire. So the higher the score, the more depressive symptoms you have. So as you can see, this red bar, it's people who don't have any family that are survivors of the Armenian genocide. They have a lower score than others who have one or multiple survivors of the Armenian genocide. So again, these rates are from studies of Holocaust survivors, prisoners of war, refugees, and other trauma survivors. So can trauma be inherited? So there's a growing body of evidence that there's a biological mechanism that environmental factors cause epigenetic changes, which are inherited by offspring. But there are some criticisms to this. Again, this is a new emerging field. So the connection may not only be attributed to epigenetic changes, the connection might be that parents who are trauma survivors have a different parenting style and behavior that children may adopt. So parents can may be neglectful or abusive, or just children are adopting their parents' behavior. Or that it can also be that children are growing up in a similar environment than their parents, like a war-torn country. Maybe the children are also growing up in that type of environment. It can also be that just that parent originally had a genetic predisposition to the effects of trauma, and so that genetic predisposition was inherited, and the child has that as well. It can also be social transmission. So if you're sitting around the table listening to your grandma, who's a Holocaust survivor, talking about her experience, you can kind of have that vicarious effect of trauma. And also these studies, they are subject to criticism because of small sample sizes. So just a few definitions. So a genotype is the genetic code of the individual gene. So the DNA is made up of a sequence, ATCG, and kind of that code is what makes up genes. And then the phenotype is the expression of those genes, of that genotype, that is visible or observed. And epigenetics is the modification of gene expression rather than the alteration of the genetic code. So it changes how your body reads DNA, and it can turn genes on or off. So as you can see in this figure, there's different ways to achieve one phenotype. So you have classical genetics, which is your genetic code. You can have changes in your DNA sequence, and it can lead to phenotype, a different phenotype, or your environment can cause epigenetic changes, which can also cause a different phenotype. So just a quick overview of kind of what we went over. So this is how DNA is packed in the body, and you can have a mutation or a change in the genetic code, and it can lead to a different phenotype. Or with epigenetics, again, you have that DNA sequence, the way the DNA is packed, and then you have little tags. So you can have DNA methylation or modification of histones, which are the proteins wrapped around DNA. So you're not changing the code itself, you're changing how the DNA is interacting, and it can also lead to different phenotypes. Okay, so epigenetic changes in offspring and trauma survivors. So this is some evidence. So the figure on the left shows Holocaust survivors and their offspring have epigenetic changes in the same place. So the x-axis is looking at one epigenetic change, which is the methylation of FKBP5, and that has been associated with cortisol dysregulation. So you see so F0 is the parental generation, so that's the percent methylation of that epigenetic change, and then you have the y-axis, which is the F1% methylation of that same gene, which is the offspring. And as you can see, there's a correlation So the red squares are the Holocaust parent and offspring versus the non-Holocaust parent and offspring. So that's a study in humans. And then kind of to confirm this even further, there's a study in mice. And it's looking at mice, control mice, and mice who were exposed to maternal stress. So kind of the trauma, the mice who were exposed to trauma. And it's showing their, there's an RNA sequencing of their sperm. And it's looking at long RNA, which, as you can see, the control mice versus the trauma mice, their offspring had different expression of RNA, of the long RNA. And some conclusions. So this is a relatively new and still controversial field. There's much more research to be done. And so the science of epigenetic inheritance of the effects of trauma is young, which means that it is still generating a heated debate. And it is important not to get kind of sensationalized in the headlines.
Hi everyone, and welcome to my presentation titled Transposons Are Jumping Genes, presented by me, Shivangi Sinha. So first off, I know most people haven't heard of transposons before. A transposon, also known as a jumping gene, is a DNA sequence that can move throughout a genome. I know that sounds terrifying, but I don't want everyone to freak out. After millions of years of evolution, most transposons are innocuous and either lost their ability to jump or can move without horrible consequences. So our genes aren't exactly like a published book, if you thought that before. Transposons were discovered in the 1940s by late Dr. Barbara McClintock in a maze model while she was investigating why different corn kernels can appear multiple colors even if they're on the same corn cob. She discovered that different colored corn kernels were caused by transposons jumping in and out of important genes responsible for corn color at different times. Now that we know that, it's important to know that there are two main classes of transposons. Class 1 transposable elements, also known as retrotransposons, are able to move or transpose through a copy and paste mechanism. Looking at the diagram, we can figure out exactly what this means. Firstly, the donor DNA is transcribed into an RNA intermediate. Afterwards, the RNA intermediate is reverse transcribed into a cDNA intermediate, which will be integrated or jump itself into the target DNA sequence. As we can see, the donor DNA is not directly transposed into the target. This is what copying and pasting means. These retrotransposons can be autonomous or non-autonomous, meaning they can either integrate by themselves or they'll need external sources to provide the appropriate enzymes to transpose. On this note, the second class of transposable elements are known as DNA transposons. This is because they do not have an RNA intermediate whatsoever the way retrotransposons did. DNA transposons can mobilize through a cut and paste mechanism rather than a copy and paste mechanism, because looking at the diagram, we can see that the donor DNA directly excises itself out of the sequence to then enter the target DNA sequence. The gap in the donor DNA sequence is usually refilled by DNA polymerase. There is no second copy of DNA segment whatsoever, hence the lack of copying and the introduction of cutting. DNA transposons are also autonomous or non-autonomous, depending on what factors are available. Now that we've learned more about these jumping genes, you have to wonder how common they are. Crazily enough, transposons have been seen in the genomes of almost every living organism noted to date. In humans, about 45% of the genome can be identified as transposable elements, with some newer studies estimating more around 66% of the genome. Don't fret, however. I stated earlier that most transposons are innocuous, and in us humans, over 99% of transposable elements have actually lost their capability to jump anywhere they want. In this graph, we can examine the relative abundance of transposable elements in various genomes by the percentage of the genome. Humans, noted at the top, can be seen with a range of 45% to 66%, with 45% kind of being more of the consensus. We can also see the just variety of life that transposons are in, from vertebrates, insects, fungi, plants, prokaryotes, and more. In one way or another, transposons are present from the smallest 0.1% to greater percentages over 50. Now, most of the time, transposons are harmless, but there are times where they can cause harm and disease. Such cases are with colon cancer. Line 1 is the name for a class 1 transposable element found within humans. In normally functioning APC genes, the genes are transcribed and translated to create APC protein, which is vital for tumor suppression. Sometimes, however, line 1 can transpose itself into the APC gene, disrupting the flow. When this happens, the APC gene cannot be transcribed and translated functionally, thus stopping the production of APC protein altogether. This mutation itself has been noted in approximately 80% of human colon tumors. It is important to remember, though, that the majority of transposons are indeed harmless. So what are some main takeaways? Overall, transposons or jumping genes are DNA sequences that can move throughout the genome. There are two overall classes. Class 1 transposable elements, also known as retrotransposons, have an RNA intermediate. Class 2 transposable elements, also known as DNA transposon, only ever use DNA. Transposons are found in the genome of almost every known organism to date, and though mostly harmless, there are instances of transposons causing mutation and disease. So I hope this presentation was enjoyable and that you learned something you hadn't known previously. Thank you for watching.
Hello everyone, my name is Jaidalis and today's scientific talk is titled Something is Not Feeling Right. Let's talk about endometriosis. And before beginning, I would like you all to know that March is the Endometriosis Awareness Month. And the yellow is the international chosen color to represent endometriosis. So some important facts. 1 in 10 people suffers endometriosis. For some context, this is about 176 million people in the world. Also, it is one of the leading causes of infertility. And I will talk about this a little more later on. And there can be a delay of diagnosis of about 7 to 10 years. This can happen because for diagnosed endometriosis, a procedure called laparoscopy has to be done by a physician, where he does a small incision in the umbilical area and the pelvic area to insert a camera and to visualize for endometriosis lesions. This graph was made by Nohaman et al. In 2011 and represents the diagnosis delay of endometriosis in different countries. We can see that the United States is represented by the state of Boston, and they have a diagnostic delay of about 8 to 10 years. Also, we can see that alongside with Sao Paulo, that is Brazil, they are the second country leading in the diagnosis delay of endometriosis. So what is endometriosis? Well, it is a chronic reproductive condition. And to better understand what is endometriosis, we must recall how menstruation happens. This graph shows us the endometrium, that is the lining of the uterus. During menstruation, the endometrium sheds due to hormonal changes. So that tissue that is shed passes through the cervix, then to the vagina and outside of the body. So if tissue similar as the tissue of the endometrium grows elsewhere of the pelvic area, then both tissues will shed during menstruation, and only the tissue that is inside of the uterus can go outside of the body. That is how endometriosis happens. What are some symptoms of this condition? Heavy menstrual bleedings. They can happen any time of the month too. Long menstrual periods. A typical period can last about 5 to 7 days on average. Painful intercourse, pelvic pain that can happen also any time of the month, and infertility. Here is an example of how infertility can happen. We have a section of the fallopian tube. Here we can see the sperms, the egg, and some endometriosis lesions. So if the sperms cannot reach the egg, then fertilization can't happen. Also, in this other example, we can see the ovary, an egg, and endometriosis lesions. So if the egg cannot reach the fallopian tube, then ovulation can happen, therefore fertilization neither. This image shows us how endometriosis looks like. The spots of red and black color is tissue similar to the endometrium that remains inside of the body during menstruation. So why do we care about endometriosis? Well, at the moment, there is no specific biomarkers leading to the diagnosis delay that we saw earlier in this talk. Lack of awareness. Although 1 in 10 people suffer endometriosis, a lot more than that doesn't know that this condition exists. Therefore, we must continue educating to stop the social stigma that people have towards reproductive health conditions. And there is no cure. Here we can see an uterus screaming, and we shall listen if a uterus is screaming. Thank you all for listening, and thank you to these amazing graduate student organizations.
Hi everyone, my name is Natalia Soto, and I am a second-year undergraduate student majoring in neuroscience at the University of Illinois at Chicago. Today I will be sharing how the gut microbiome is our second brain and why that is important. So first, what is the microbiome? Well, the microbiome is the largest collection of microorganisms in our bodies found in the gut. It consists of trillions of microbes like bacteria, viruses, and fungi that make up the gastrointestinal tract, which is the gut. And a fun fact about the microbiome is that it contains more genes than its human host because of the amount of microbes that live in it. Now, the gut microbiome is known as our second brain because of its importance and its ability to operate independently from the central nervous system, which is the brain and the spinal cord. And this makes up something called the enteric nervous system. The microbiome's ability to operate independently from the brain is in great part due to the fact that after the brain, the gut contains the largest amount of neurons in the body, with there being approximately 500 million neurons. And here in this picture, you can see that depicted in the color pink. Even though the enteric nervous system, and therefore the microbiome, are able to work independently from the central nervous system, our microbiome and our brain still engage in a bidirectional communication that flows both ways through something called the gut-brain axis. The gut-brain axis serves as a bidirectional communication network between the central nervous system and the enteric nervous system. And studies have found that the gut microbiome plays an important role in influencing the interactions between these two systems. Gut microbiome health can have a considerable impact in the gut-brain axis. And given the bidirectional relationship this axis has with the central nervous system and the enteric nervous system, this impact is not only observed locally in the gastrointestinal system, but also in the brain as well. This relationship between the gut microbiome and the gut-brain axis suggests that changes in the gut microbiota can alter molecular mechanisms in the brain and therefore inflict changes in behavior too. As you can see in this image, the microbiome can alter neurotransmitter release in the brain, which can then have an impact in our behavior, our mood, and our stress levels. On the other hand, the brain can have an impact in the gut by altering motility, which is the contraction of our digestive muscles, on secretion, which is waste removal, nutrient delivery, and even it can even alter microbial balance, which can then lead to digestive illnesses. Now, what do we care? Well, mental health is a highly prevalent issue in the United States, across all races and ethnicities, as can be seen on this graph. About 20%, which is 1/5 of the US population, suffers from mental illnesses. Because of this, there's been a renewed interest in trying to understand the underlying mechanisms of mental disorders in the hopes of finding new treatments and therapies to alleviate the symptoms of these illnesses. As a result, and given the relatively new knowledge of the gut-brain connection, numerous researchers and experts believe that studying the microbiome in relationship to diseases like depression and anxiety will help us understand the underlying mechanisms and potential treatments. One of the most common digestive disorders that affect the gut is irritable bowel syndrome, or more commonly known as IBS. So numerous studies have been performed evaluating the relationship between mental health and this condition. As you can see on this graph, there is a correlation between mental health and IBS. People that suffer from IBS, which are represented in the blue bars in the graph, experience mental disorders like depression, dysthymia, which is another kind of depression, panic disorder, and substance abuse at greater percentages than the general US population, which is represented in orange. And that means that you are more likely to suffer from mental health issues if you have IBS than if you don't. Other research has shown that gut microbiota can regulate psychological disorders. There was a study that found that the vagus nerve stimulation, and the vagus nerve is the organ that carries out the gut-brain connection. If you stimulate the vagus nerve, it's been shown that you can improve depression symptoms. Another study also found that chronic gastrointestinal inflammation induces anxiety-like behavior, and these research provides evidence for the existence of the relationship between the brain and the gut. There was another study that was done that found that probiotics, which are supplements that promote good gut health, they have a positive effect on mental health, which shows that encouraging potential for the development of new mental health treatments. Now, finally, I would like to share a quote from microbial ecologist Jack Gilbert, because I feel it captures the importance of studying the gut-brain connection given the high prevalence of mental disorders. He says, "Within 5 years, I hope to see more clinical trials that demonstrate the efficacy of prebiotics and probiotics on mental health disorders. There needs to be a revolution in how we deal with mental illness in our society, and I couldn't agree more. This revolution needs to be inclusive, and tailoring research towards our second brain in relation to mental health is a great way to start." Thank you.
Good evening, ladies and gentlemen. My name is Samuel, and today I'll be talking to you about smart mobile systems development for monitoring and helping patients suffering with chronic diseases. Do we know how many people living with chronic diseases around us today? 1 in 2 adults in this country has a chronic disease. 1 in 4 of these adults in this country has 2 or more of these diseases. Now, on a global scale, we have more than 50% of the world that lives with a chronic disease. There are several examples of these chronic diseases. Examples are the heart failure, blood pressure, diabetes, etc. With people living with these chronic diseases, there's the need to monitor their activities and guide their lifelines. To do this, there are several applications that are programmed in helping these people manage their condition. Due to limited time, I will focus my talk on one major chronic condition, which is the heart failure, and how the heart failure Storyline smartphone application could be used in managing it. Heart failure is a chronic condition where the heart does not pump blood the way it should. People who have this kind of condition are associated with symptoms such as the chest pain, severe weakness, and rapid or irregular heartbeat associated with shortness of breath. This report shows the lives lost per 100,000 rate adults suffering from heart failure disease in the year 2014 to 2016. The race was calculated by looking at the number of risk factors, including high blood pressure and cholesterol, and smoking habits, obesity, and low consumption of fruits and vegetables. The results showed a remarkable variation across the state. People with a higher heart rate disease risk tend to live in the southern state, as it shows on this side. The people suffering with this kind of condition could be helped with smartphone applications like the Heart Failure Storylines app. The use of this app helps patients in supporting their condition, and it also combines features such as guidance and communication to enable these patients use the application in managing their condition by filling out different survey forms. Practically, the application has several features that accept data input from the patient and displays results according to the data it receives. For example, using the symptom tracker feature, which allows the patient to enter symptoms such as headache and weakness, and it also allows you to rate your symptoms on a scale of 0 to 10, as you would do when you go to the hospital to tell how much you feel these symptoms. The result is then displayed on the screen graphically according to the data that it received from the patient. And the peaks shown in this diagram shows the intensity at which the patient feels that symptom. This saves the patient a lot of time, and it makes them aware of their condition to see if they need further treatment or attention. It does not end here. People suffering from other chronic conditions or diseases could be helped by using other smartphone management apps such as these to carry out symptoms, to carry out symptoms, to carry out similar guidelines in monitoring their health condition. The intellectual merits of this study is that compared to the traditional method used to monitor health condition, mobile health applications will reduce health costs, it would enhance the efficacy of patient care, and the broader impact is that this mobile health applications will increase public understanding of health conditions by placing real-time monitoring of health condition in the hands of any patient that had access to a smartphone. It would also more than likely reduce the burden of emergency visits to the hospital, positively impacting the overall quality of life of patients and society as a whole. Overall, to be able to enhance the efficacy of patient care, the goal is self-management. Thank you.
Hello everyone. Today I will be discussing ultrasounds, which are a medical imaging technology that is used for checkups and diagnosis. Some of the things that I will be discussing in my presentation today are what is medical imaging, what is an ultrasound, common uses of ultrasounds, benefits versus limitations, and some of the future potential of this technology. So what is medical imaging? Medical imaging is a process that refers to techniques that produce different images of the body for diagnostic and treatment purposes. There are various types of medical imaging, such as X-rays, CT scans, MRIs, and PET scans, and of course, ultrasounds. What is an ultrasound? Ultrasounds can also be referred to as sonography, and it's a procedure that uses high-frequency sound waves to produce images of the part or the organ being scanned. It uses a small probe called the transducer and gel to allow sound waves to move into the body. It captures images in real time, and therefore it can show movement and blood flow. Some of the common uses of ultrasounds include checking on fetus in pregnant patients, scanning brain, hips, and spine in infants, checking on the liver, gallbladder, spleen, and pancreas, which are very important organs in the abdominal area. It can also help diagnose heart conditions because it could show movement and blood flow in real time. It can also be used to guide procedures such as needle biopsies. Some of the benefits versus the limitations of this technology include the benefits are that it's non-invasive, there is absolutely no risk to the patient, it is not painful, and it is widely available and inexpensive. Moreover, it can show clear images of soft tissue that are not clear with X-rays, and it can provide us real-time images with motion. Some of the limitations include that the images can be disrupted by air or gas, and that is not useful for imaging in the lungs because there's a lot of gas exchange. And more limitations include that it is difficult to visualize tissue in larger patients because the sound waves are weakened due to having to infiltrate more tissue. Future potential of this technology, there's a lot of it because providers are now preferring to subject their patients to less radiation. So One thing that I found pretty interesting is incorporating advanced GPS needles in ultrasound biopsies, which allows for more accurate results in a much less time. And fusion imaging, which is combining images from two modalities to get the best possible picture. This is already widely available in terms of combining CT with PET scans and MRIs with PET scans, and that has been very, very useful for doctors and patients alike. So doing so with ultrasounds can be very, very helpful as well.
Hello, everyone. My name is Yuzu, and I am here to share a study investigating the interplay between obesity, cancer, and the immune system and give you a warning. Obesity weakens your immune system's ability to kill cancer. So first of all, if you think about a tumor inside a cancer patient, a tumor is not only composed of tumor cells, but also immune cells, blood vessels, stroma cells, and make up this unique environment that affect how a tumor can grow or spread. You can relate this situation as to a small evil village that may appear in some kind of fairy tales, with perhaps castles, roads, people inside the village, such as king or queen, that want to expand their territory and invade into big cities. Now, there is a key character in the immune system who works to fight against the evil village, an immune cell called the CD8 T cell. CD8 T cells are able to recognize the tumor as a target that needs to be removed, and they're actually able to kill the tumor cell. We can refer them to the dragon, like presented in this figure. CD8 T cells are the main weapon used by the immune system to fight against the tumor. CD8 T cells are the central focus of many promising precision cancer therapies. However, in order for them to fight, of course, like most of us, they need to have sufficient energy to kill the enemy tumor cells. Next, speaking about obesity, our central focus today, obesity is a chronic illness resulting from multiple environmental or genetic factors. Fat cells may be referred to as soldiers who come to help sustain every cell in the body to give them energy. However, in the situation of obesity, there are just too many fat cells for the cells to use as energy resource, and as a result, keep on sustaining the evil village tumor with energy resource for them to grow and expand their territory. Speaking of obesity, they're often associated with poor immune responses. But here's the question. If the soldiers or fat cells are sustaining fuel for the bad village, wouldn't the immune system also get the same amount of energy as well? What about the weapons? If they are getting energy as a whole, why can't they fight anymore? To answer these questions, researchers compared the mice fed with controlled diet, normal diet, and high-fat diet, in which they were injected with the same number of tumor cells and observed how fast the tumors grew between the two groups. Looking at this figure as a result, tumors grew faster with high-fat diet given mice. Now, why was it that high-fat diet allowed tumors to grow? It was suggested that fat cells are somehow removing the dragon, CD8 T cells, from fighting against the tumor. Surprisingly, when the researchers compared the tumor from the mice on a normal diet and high-fat diet, the number of CD8 T cells were reduced. Here, tumor cells are shown in blue and CD8 T cells are shown in red. When we look closer at this part, you can see that the presence of CD8 T cells are reduced in the tumor microenvironment given with high-fat diet. Researchers found out that obese, fat-rich environment changes the way tumor cells take up fuels, making them get energy efficiently and able to grow and expand their territory. On the other hand, because tumor cells keep on taking up the fuel, CD8 T cells become starved and unable to get energy for them to grow. As a take-home message, a high-fat diet reduces the number of CD8 T cells and increases tumor growth. This is because in condition of obesity, the tumor cells are much better at stealing energy, and CD8 T cells do not have enough fuel for them to function. Therefore, CD8 T cells cannot control the tumor anymore. This is the reference I used to present in this study. Thank you all for listening, and I hope you all have a wonderful day.
Hey, hi, everyone. My name is Kiara Brown. I'm an undergraduate student at Georgia State University at the Institute of Biomedical Sciences in Atlanta, Georgia. And today we're going to discuss the health effects of disinfectant byproducts in drinking water. Okay, so you guys may remember when you were in your general chemistry class and your professor told you that the molecular structure or the molecular formula of water is H2O. But one thing that your professor may not have told you is that our drinking water consists of a little bit more. Our drinking water can consist of pathogens, yeah, that's right, chlorine byproducts, nitrates, pharmaceuticals, and so much more that I haven't even listed on the slide. You'll be surprised. But that's all to say that water is often needed to be disinfected before we can consume it. Now, there was a time in the not so distant past where we didn't have a proper water purification process, and people were dying. People were dying from a different epidemic caused by a deadly bacterium known as cholera. But this all changed by the 20th century when chlorine was discovered by a chemist named Carl Sleet, and when chlorine was started to use as a water disinfectant. So this changed everything. It revolutionized the water purification process by killing harmful bacteria, such as cholera, like I mentioned earlier, but this didn't come without consequences. And one of the consequences was the formation of disinfectant byproducts, or DBPs, in our drinking water. Now, this all arose from a public health crisis back in the 70s, where it was discovered that there was more than 100 disinfected byproducts in our drinking water. And it was also discovered that these disinfected byproducts could be toxic and even carcinogenic. Now, you may be wondering, how does disinfected byproducts get in our water? And what is a disinfectant byproduct? Well, a disinfectant byproduct is simply a chemical byproduct found in our drinking water that's basically formed from a water disinfectant, such as chlorine, reacting with other naturally occurring organic matter. And there's one thing that you need to take away, that these disinfectant byproducts are very difficult to remove from water. So to give you guys a better illustration of how disinfectant byproducts get in our water, we're going to focus on the image here in the middle of this presentation. And basically what happens is chlorine, water disinfectant, is added to our drinking water. It forms hypochlorous acid, and hypochlorous acid then reacts with bromine or iodine from the naturally occurring organic matter in our water pipes. And what happens is hypochlorous acid then turns to either hypobromous acid or hypoiodic acid, which then forms into these disinfected byproducts that are very difficult to remove. Now, there's two classes of disinfected byproducts. There's the trihalomethanes and haloacetic acids. But today we're going to focus on the trihalomethanes because there's more research focused on trihalomethanes. And right here on your right, there's an image of a trihalomethane. It's basically a carbon atom with 3 to 4 halogen groups attached to it. Now, it looks perfectly harmless here, but let's not be deceived. Okay, so that begs the question, how does disinfectant byproducts, or DBPs, in our drinking water affect our health? So that's a good question. Now, since the 1970s, there have been multiple studies done on connecting DBPs with some really nasty health illnesses, such as increased risk for bladder and colon cancer, miscarriage and birth defects, liver and kidney damage, and even cardiovascular defects. But we're going to focus on a study that occurred in Iowa. And it basically focused on postmenopausal women and the drinking water that they were exposed to in the state of Iowa. So over 28,000 participants participated in the study, and it was limited to people who drank private well water, mixed water, or surface water / tap water. But what was significant about this data was that it was found from this graph here, it was found that surface water or tap water, which is shown with this gray line, the women that drank the surface water or the tap water were at a higher risk for colon cancer, and that's highlighted here, than the women who drank either private well water or mixed water. And in the same study, another focus was on a disinfectant byproduct known as chloroform and its increasing levels of exposure. So again, not as many people participated in this study. It was like 16,000 people participated in the study in the state of Iowa. And chloroform, like I said, it's a trihalomethanes. It was a variable of primary interest. And what was significant about this data is that as the increasing level of chloroform was in their drinking water, they were not only at risk for colon cancer, but at a high risk for melanoma and lung cancer. And these markers here, the one up here shows the one for melanoma, this is for lung cancer, and this right here is for colon cancer. Now, the huge takeaway that I want you guys to take is that water isn't simply H2O. Our water has to be disinfected, like I said before, and it's primarily in the United States disinfected by chlorine. And that means inevitably our water is going to have disinfected byproducts such as trihalomethanes and haloacetic acids, which means that studies have suggested that although there's a high correlation between disinfected byproducts and cancer, we still choose to disinfect our water with chlorine, and that's because it's generally assumed that that exposure to pathogens in our water has a worse health effect than exposure to disinfectant byproducts such as trihalomethanes and haloacetic acids. But the reason why I brought this discussion today was because there's two things that needs to sustain life on our planet, and that's the sun and water. And everybody should have access to water in order to continue life as we know it. So I encourage everyone to continue this discussion and to look further more in research, so that we all can have safe, clean water today, tomorrow, and in the future. And that's my presentation. Thank you.
Hello, everyone. My name is Emily Carter, and I'm excited to share this important topic with you that's relevant to today's society, the relationship between obesity and severe COVID-19. And so it's no secret that COVID-19 has impacted the world. Many people have died from this disease, and specifically in this graph, you can see the cumulative total number of people who have died in the United States. And the darkest states here present the states with the most COVID-19 deaths. And you can see the distribution of color. The lightest states have the least number of deaths. But overall, the entire US has been impacted. But what a lot of us don't know about COVID-19 is how people with chronic illnesses have more severe cases of COVID-19 infection. And so having chronic illnesses that people live with, and some examples are obesity, diabetes, cardiovascular disease, which is abbreviated as CVD here, people are more likely to have severe forms of COVID-19. And overall, they're more likely to be admitted to the ICU, which is the intensive care unit, and overall have an increased chance of dying from COVID-19 infection. And so a US study found that obese patients in particular are at a 1.31 times higher risk of ICU admission compared to patients who are not obese, and as a result have a greater risk of dying. And so what are the risk factors? So here you can see on this graph, this presents hazard ratios, which is a statistical number that tells you how much more likely you are to die from a specific disease or condition. And it's based on different factors. And so here we are looking at the BMI, which is highlighted in this box. And BMI stands for body mass index. And so as we can see, having a BMI of greater than or equal to 40 leads to a greater risk of COVID-19 or dying, because values on this graph that are greater than 1 indicates having more severe COVID-19 or to die. And compared to other BMI values, you can see here they deviate from 1. Having 1 greater than or equal to 40 is furthest away from the 1 value. And so some other factors you can see at the bottom of this graph, like chronic heart disease, liver disease, stroke, and dementia. These are other chronic diseases that show the same trend. And so what is the relationship between obesity and COVID-19? Well, we can see in the data that there is a trend there. Having obesity leads to greater risk of having severe COVID-19, but overall it's a public health issue. So how can we reduce the death toll by curing obesity? And what happens in the future if there's other respiratory viruses that have the same death toll or rate? And so what we've learned from the data is that we have to encourage changes in diet and exercise in order to achieve a healthier lifestyle. And this involves educating the general public about healthy diets, how to read food labels, what exercise to participate in, when to exercise, what to eat, and overall develop long-term healthy behaviors and habits. And so we have to start now. We know that obesity leads to more severe COVID-19 disease. But what can we do? We can take action by creating a healthier lifestyle for ourselves and for our families. And as a result, all of us can be a part of the solution in reducing severe COVID-19 outcomes, because the COVID-19 pandemic is still happening. And in the future, we don't know what will happen there with other viruses, other diseases. So we all have to be a part of this solution. And that is my presentation today, and I hope you learned more on the relationship between obesity and COVID-19.
Good evening, my name is Adriana Rivera Rodriguez, and my presentation is titled Fecal Microbiota Transplant as a Treatment for Gastrointestinal Symptoms for HIV Infection. So I want to talk briefly about how do fecal microbiota transplant work. A fecal transplant is a procedure that transplants stool or poop from a healthy donor to a patient via an oral capsule, otherwise known as a poop pill. Fecal transplants restore the patient's microbiome. Think of your microbiome as the bacteria in your gut, the healthy kind of bacteria. So due to this very property, it is currently being studied as a potential treatment for HIV symptoms. So let's talk about HIV. HIV is a disease that attacks the immune cells, especially the ones concentrated in your gut. These cells regulate your natural microbiota. And when your natural microbiota is disturbed, you can experience an array of symptoms, these being inflammation, constipation, diarrhea, nausea, vomiting, or weight loss. Whether you have HIV or not, if you experience a disturbance in your natural microbiota, doctors will probably prescribe you probiotics, which are live bacteria that have beneficial effects for digestive symptoms. You could find probiotics in foods like yogurt, or you could get them as a supplement. You can think of a fecal transplant as a probiotic treatment. So let's talk about the actual treatment for HIV. These are antiretroviral drugs, which are the leading treatment for HIV since there's not a cure or a vaccine available at the moment. They work by stopping the HIV virus from replicating. However, antiretrovirals do not replenish gut health. In many cases, they can worsen it. It really will depend on the mix of antiretroviral drugs that the patient take, but they could cause vomiting, diarrhea, and or nausea. So what's the difference here? HIV patients have to take these drugs in order to stop the HIV virus from replicating. However, they're still experiencing the same uncomfortable symptoms, and this causes the HIV patients' quality of life to be very poor. So right here, what I'm showing is a graph. The x-axis represents time in weeks, while the y-axis represents total CD4 plus T cell count. These are immune cells, and these shapes represent the subjects, which were macaques, our species of monkey, that were put through different procedures, which I'm going to talk about very briefly. First, we have SIV, which stands for Simian Immunodeficiency Virus. It works very similar to SIV, and this represents like a typical immune cell count for a subject that has SIV. Then you have ART, which stands for antiretrovirals that we talked about before. And as you can see, there is a very dramatic drop here in the immune cell count. Then we have ABX, which stands for antibiotic treatment. And this was administered just to give the fecal transplant a clean slate and to kill off any pathogenic bacteria that might be like lurking in the subject's gut. But you can see a slight increment in the immune cell count. Then finally, we have the FMT, which stands for fecal microbiota transplants. And you can see a significant increase in the immune cells. What am I trying to say with this graph? The FMT is capable of increasing the immune cells, therefore stabilizing the natural microbiome and relieving some of the symptoms that HIV patients experience. So what is the takeaway from this? Fecal microbiota transplants could potentially be a safe form of treatment to restore gut health in HIV patients, therefore increasing their quality of life. Also, fecal transplant could be used for individuals who do not have HIV but have digestive problems that are similar to those with HIV. So this was my presentation. Thank you so much for your attention, everyone.
Hello, everyone. My name is Karima Brown. I'm an undergraduate student at Georgia State University studying biomedical sciences and enterprise. And today we're going to be discussing the complexity of schizophrenia, or as I have termed it, the rabbit hole of schizophrenia. Schizophrenia has gone by many different names. To name a few, in the past, it has gone by adolescent insanity, catatonic syndrome, hyperphonia, and it was categorized as a disease with an unknown cause, one that mainly afflicted the youth and led to chronic deterioration of the mind. Schizophrenia today still remains underneath the same cloud of ambiguity. It is classified as a broad clinical syndrome, one that is based on subjective experiences, loss of function, and the symptoms vary from patient to patient. What I would like for you all to take away from this presentation is schizophrenia is not synonymous with insanity. So let's take a look at the origins of schizophrenia. It all started in 1893 with Emil Kraepelin. He gave the first name for the disease. He called it dementia precox. And he said the disease was a unitary disease, meaning that it was a disease of a single entity, but with various conditions. While his work was groundbreaking for the time, it did not provide any diagnostic criteria. So in 1908, Eugene Buehler gave the disease a new name, one we're very familiar with, schizophrenia. He said, unlike Kraepelin, that it was a divided disease, meaning that it was a group of diseases. Kraepelin and Buehler's work stands as the foundational blueprint for how we view schizophrenia today. However, it's an ever-evolving process. To make my point, today we use the Diagnostic and Statistical Manual of Mental Disorders, or DSM for short, to diagnose all mental disorders, including schizophrenia. In 1994, we used the fourth edition, where it was based off of an actual system and it classified schizophrenia into different types. Today, we use the fifth edition, which did away with the actual system and the different types. Overall, DSM has greatly improved our accuracy of diagnosis of mental disorders, including schizophrenia. However, it wasn't until the fifth edition that we began to appreciate schizophrenia as a spectrum disorder. This is important to note because our diagnostic tools play a critical role in the management and treatment of schizophrenia. To illustrate my point, in a study focused on the prevalence of schizophrenia in high-risk individuals, as seen in the y-axis here, DSM-4 was used to diagnose these individuals aged 6 to 7 years of age in the x-axis here. But what I would like for you all to focus on is the pie charts, which show that individuals in the younger age groups were more likely to be diagnosed with psychotic disorders not otherwise specified, as can be seen in the orange or yellow color. Whereas older individuals in the older age groups, 26 years of age and older, were more likely to be diagnosed with schizophrenia, as shown in the blue here. This is important because this is the reason why the diagnostic manual was updated from the 4th edition to the 5th edition, due to the prevalence of overlapping symptoms and low diagnostic precision, meaning that any of the individuals in the younger age groups could, go their whole life without receiving treatment, proper treatment. So, as I said in the beginning of my presentation, schizophrenia is not synonymous with insanity. To say otherwise is misleading because it can undo all of the work that has been done thus far to understand the disease, and it can lead to misdiagnosis, as I showed you in the previous slide. Schizophrenia is a very complex disease. It has a spectrum of related disorders, and that can range from delusional disorder to schizoaffective disorder, just to name a few. This now more than ever is prevalent due to the COVID-19 pandemic, where the discussion of mental health has become increasingly prevalent. So I encourage everyone to continue this discussion, and thank you for your time.
Hi everyone, my name is Nicole Felix, and today I will be talking to you about inflammation and whether it is a friend or a foe. That is to say, whether it is beneficial to you or not. Here, we have some white blood cells, the main drivers of inflammation. Let's dive in. When a wound swells up, turns red, and hurts, it may be a sign of inflammation. Generally speaking, inflammation is the body's defense or immune response to an irritant. An irritant is any foreign object that enters your body. It can range from a bacteria, a virus, to a splinter. When an irritant enters your body, white blood cells are recruited to the site of injury or infection and release various substances called inflammatory mediators. These inflammatory mediators cause the small blood vessels near the injury to widen and allows for more blood to flow to the wound site. This is why inflamed areas turn red and feel hot. The increase of blood flow allows for the friendly cells of the immune system to arrive at the injury site, where they help with the healing process. Unfortunately, inflammation doesn't always work how it's supposed to. Sometimes, instead of protecting you, your immune cells start attacking your own healthy organs, causing injuries on your healthy body. For example, let's say you get sick with the flu. The virus sets up camp in your lungs, setting off signals that leads to inflammation of the infected lung tissue. When the cells come in, they attack the virus, but they also attack healthy lung tissue due to excessive inflammation. In this case, your white blood cells stop being friendly and start acting like villains. Let's take a closer look at these health complications. A common complication of the flu virus is acute respiratory distress syndrome, or ARDS. ARDS is lung damage that occurs because of the inflammation that is persistent in the organ, even after the virus has been neutralized by the immune cells. As mentioned before, this is because the immune cells cannot stop and they continue attacking healthy tissue. This causes serious damage to the lungs, and instead of our friendly blood cells helping the lung heal, the villainous cells actually cause damage and scarring in the lungs, which if left untreated can lead to organ failure. The figure shown here is a microscopic image taken of a lung tissue infected with the flu. The top two images show healthy tissue, and this red circle points to a magnified image at scar damage on an unhealthy tissue that has ARDS. Let's recap. Inflammation is your natural, friendly response to fight off infections, and although it may hurt, it's supposed to be helpful. Like many things, it is bad for you in excess. When your body cannot regulate it well, that's when health complications occur. Lastly, scientists are looking at ways to control inflammation with medicine in order to prevent any complications. Thank you so much for your attention, and I hope you enjoyed learning more about your body.
Hi, my name is Siobhan Powell. I attend Georgia State University, and I'm a YBIDC program scholar, and today I'm going to be presenting on the evolution of the third molar. Wisdom teeth, or the third molar, has been an important part of our hominin evolution for millions of years. Going back to the genus Australopithecus to our current genus Homo, there have been many evolutionary changes, not only with our bodies and locomotion, but also with our teeth. This change, albeit slow, has been very important and has a lot of implications about our evolutionary past and also our future. There are many theories as to why and when these changes have occurred, but there's not specifically one answer. There seems to be a myriad of connected reasons, but I'll be focusing on third molar agenesis and its relation to diet. And here at the bottom, I just have a figure that kind of shows like an evolutionary timeline, just depicting the changes in craniofacial size that have happened over the years. So that'd be nice to keep in mind for reference throughout the presentation. And so agenesis simply defines when something is absent, and the main reasons for third molar agenesis seem to revolve around overall changes in diet, developmental delays, and craniofacial size. And specifically focusing on diet, when we think of how our diet has changed over time, it makes sense that there would be some sort of impact on our teeth. But another way to look at the impact of diet is to examine the extensive tissue hypothesis. This diagram depicts how a lot of energy is needed to have a large brain. So having an easier to digest diet, and it reduces energy towards digestion, and that diverts more energy towards brain growth. This lends to the idea of that the trade-off for a larger brain was less prognatism, which left less room in the mandible and maxilla for larger teeth. And one interesting thing to point out is that third molar agenesis seemed to have begun around 1.5 million years ago when Homo erectus was present. But compared to earlier hominins in the genus Homo, we can see that their diet consisted of more dense, fibrous, and tough foods, which meant that they had a need for several large, strong enameled molars in order to properly masticate and ingest their foods, which began to change in Homo erectus. Because of the gradual change in diet, that included an increase in meat eating and a decrease in tough fibrous foods that led to a change in brain and craniofacial size, which then led to a change in molar acquisition and size. There are a couple examples of Homo erectus that show M3 agenesis. For example, Laonesian man found in China, and also the Turkana boy who was found in Kenya. And here you can see that the dentition is 2122 versus the 2123 dentition that exhibits the third molar. And you can also see that there's not a lot of space in the back of the mouth for there to even be a third molar here. So that kind of shows that third molar acquisition was not at all homogeneous within the species and seems to show a major evolutionary shift then that has persisted until today. And for modern humans, the advent of agriculture and the Industrial Revolution signified an increase in diets that consisted of soft processed foods and also a lack of diversity in diet. And one function of the third molar is to take the brunt of the wear of the three molars. But with modern humans, since there's not much wear or use with the third molar, the evolutionary pressure to sustain large teeth and molars has lessened. And it seems like people either experience impaction and have to have their teeth pulled, or they experience agenesis. And this figure is from 2015, and it examines the predictors of third molar agenesis. And here they examine the results from 92 studies from multiple regions and found that the average rates of agenesis globally was 22.63%. And here the chart plots the frequencies per region and finds the average rate per region. And as you can see here, and the gray dots represent like each different study. There are many factors that are involved in the development of third molars, as explained before, like diet, craniofacial size, and developmental delays. But we should also consider how trends in globalization and changes in the global diet can have an impact on acquisition of the third molar in our future. All right, so there are many factors involved in influencing third molar agenesis, but it's important to consider why this is happening and what that says about our future and the evolution of our species and the viability of the wisdom tooth. So is the wisdom tooth becoming more like a vestigial structure? It may be too soon to know for sure, but it seems like some people are doing just fine without them. But to me, wisdom teeth seem to be on their way to becoming obsolete. And I hope I was able to convince you of this and explain third molar agenesis effectively. And here are my references, and thank you all for your time.
Hello, my name is Kekelezo Makau, and I'm going to be giving a presentation on how science is the data of policy. Science is the data of policy. We define science using the Science Council's definition, which states or positions science as data producers of data evidence which is used by policymakers. The players in this field in the fields of science and policy, of course, are the scientists, researchers, and public policymakers, which are politicians, public servants, and the likes. Data, as mentioned before, is the mutual friend between science and policy. Scientists produce the data, policymakers need the data, and this is a mutually beneficial relationship because the main beneficiaries in this instance ought to be society and the environment, the preservation of the environment. Which brings us to our case study, the Clean Air Act of 1963, and of course its successors, the Clean Air Act of 1970. We're going to be using a case study of how data was used to formulate this policy, but moreover, of what kind of data do we have to prove that this policy implementation has been successful in the United States. So the role players really which contributed to the implementation and the development and implementation of the Clean Air Act of 1963 and its successors are firstly the environment, because of the state of the environment at the time, which caused alarm and concern, resulting in people getting sick because of the killer fog at the time, the industrial era. People got sick and public health facilities were overwhelmed with sick people, and science had the data, science had the facts, and therefore it was beneficial that science provided that information in order to assist policymakers make evidence-informed decisions regarding how regulating air quality, regulating, trying to produce or trying to curb the problem of pollution. So science being the art of data. We're using this diagram from the New York Times of 2019 as one of the indicators to measure the data that was used in informing this policy. The particulate matter, or characterized as PM 2.5 in scientific terms, has been established by the Environmental Protection Agency of the US to be set at 12. So we have here in the x-axis the number of years from 1980 to 2017, and we have in the y-axis the cubic meters, the micrograms of pollution measured in cubic meters. So currently, the current national standard for the USA is stands at 12, And experts have said that since the implementation of the Clean Air Act in 1970, there has been improvement in the quality of air in the US. So what we're looking at now is a visual really explaining to us or giving us an idea of how the density of pollution has decreased from 1985 to 2015. Above are estimates of the pollution, the particulate pollution in micrograms per cubic meter. So from 1985 to 1995, we can see that already changes were visible. And this data shows the successful policy implementation by most states within the US that were able to implement the policy to try and keep the PM 2.5 at 12. So we can see that 2015, the density decreased significantly. We can almost assume that it was at levels below 10, which is good. So in conclusion, data at the center, with data, having data at the center of policymaking, and it being really the mutual friend between scientists and policymakers, now more than ever, we need more environmentalists, more advocacy groups, more public people being engaged more with science in order to make evidence-informed decisions as policymakers, as civilians, in terms of how we take care of the environment. Basically stop littering and many other ways in which the environment can be preserved. So we need scientific data to be made publicly available. We need to be engaged in it in order for us to make better informed decisions for us, our health, and the preservation of our environment. And this concludes my presentation for today, that science is the data policy needs in order to benefit society. Thank you for your time.